Hello, this is our presentation on hydroxyapatite nanoparticles used for gene therapy. So the nanoparticles used in this article are hydroxyapatite nanoparticles, or NHAPs. These are made of calcium phosphate, which is the same mineral that makes up our bones and teeth. But unlike gold or silver nanoparticles, which we typically use in our lab experiments, hydroxyapatite is biocompatible, biodegradable, and it's non-toxic, which makes it ideal for gene therapy. So each nanoparticle is extremely small. It's between 20 or 100 nanometers, which allows it to enter the cells efficiently while still carrying genetic material. So the researchers tested several different shapes, including here they are, sphere-like, rod-like, plate-like, and needle-like. And they use these to see how the different geometries may affect how well genes are delivered into the cells. So these nanoparticles, they bind to nucleic acids due to the attraction between calcium ions in the hydroxyapatite in the nanoparticle and the phosphate that's in DNA or RNA. And once they're inside a cell, they dissolve the acidic environment of the endosomes, releasing their genetic material where it can start acting and doing its thing. So the paper also discusses a few ways to modify the nanoparticles. As we can see here, they can be doped with ions such as um, silicon, magnesium, or zinc, right, to change how fast they may dissolve. And our adding iron actually makes them magnetic, so they can be guided with magnets through the treatment site, right? And so this image right here is actually it's what's called a transmission electron microscope. And it's what they are using to show the particles so we can clearly see the differences in shapes and sizes. Um,
Howdy, my name is Blake Henderson, and this is my presentation on hydroxyapatite nanoparticles and how we can apply them today. Hydroxyapatite nanoparticles, or NHAPs, are being developed as safe, non-viral carriers for gene therapy. Traditional viral delivery methods can trigger immune reactions and are very expensive, which this would help to prevent. NHAPs are made from the same mineral as bones and teeth. They are biocompatible, stable, and non-toxic. They protect fragile DNA or RNA and help them enter cells to repair or silence faulty genes in the body. Researchers are already using NHAPs for targeting cancer cells, to promote bone regeneration, and to support tissue repair. This research is significant because it could make gene therapy much more safe, much more affordable, and much more readily available to the public. It offers new hope for treating disease and improving healing in ways that directly benefit all of society. Thank you.
Hello, it's Georgie, and I'm building off of what Blake was just talking about, and I'm going to tell you exactly how hydro hydroxyapatite does that. The first thing is binding and loading, and basically negatively charged DNA or RNA molecules attached to positively charged surfaces of modified NHAPs through electrostatic infractions or co-precipitation. This formed a nucleic acid complex that protected the fragile genetic material from enzymatic degradation in the bloodstream. The second thing is cell entry. Cells absorb the complexes through endocytosis, an energy-dependent process where the cell membrane folds around the nanoparticles as it draws it inside. This was confirmed visually through fluorescence and TEM imaging. The third thing is endosomal escape. Once inside the cell, the nanoparticles were trapped in acidic vessels called endosomes. Hydroxyapatite is pH sensitive. It dissolves in low pH environments, releasing the calcium and phosphate ions. These ions cause the osmotic swelling and rupture of the endosomes, freeing the genetic material into the bloodstream. The fourth is gene action. For RNA therapies, the released RNA entered RNA-induced silencing complexes to silence targeted genes. For DNA therapies, plasmid DNA traveled along microtubules into the nucleus, where it was transcribed and translated into therapeutic proteins. Lastly, it was just surface modifications for efficiency. Researchers improved delivery using coatings like polyethylene or polyargentine, which made nanoparticles positively charged and helped endosomes escape via the protein sponge effect.
All right, so these nanoparticles actually have some really crucial improvements on some current technologies right now. So in tumor therapy, you can see this where there's a lot of potential treatment cases for many types of tumors, such as hepatoma carcinoma, breast carcinoma, prostate cancer, pancreatic cancer, glioma, etc. These nanoparticles have actually proven to be more effective than just normal tumor targeting drugs due to their lack of cytotoxicity on normal cells. Because obviously, in these situations, you want to focus on just killing off the tumor cells while leaving normal, healthy cells intact. Um, and then in bone regeneration, you can see this where the nanoparticles can actually be used as a coat for implants in order to improve their osteogenic activity, just allowing these bone cells to grow and develop. And you can see that synthetic hydroxyapatite blocks have actually been found to have inadequate fracture toughness. So they're actually unable to be used in any kind of load bearing defects, whereas the nanoparticles actually increase the degradation rate from material to bone.
Just to finish off, this is our citation for the article we used. Thank you all for listening to our presentation on gene therapy and how hydroxyapatite nanoparticles help with that, and being able to harness that for many different uses in the gene therapy space.